Hello, everyone. Thank you for joining us today for this joint webinar. This webinar is the 12th in the series on the use of NOMs in risk assessment and the 7th in the neurodynamic discussion series. The webinar is being co-organized by the Physicians Committee for Responsible Medicine, the U.S. Environmental Protection Agency, the PETA Science Consortium International, and Unilever. I will give a second to let more people log on. I see we have some participants coming in. Feel free to say where you're coming from in the chat. Welcome, everyone. We're glad to have you. And my name is Bridget Rogers. I'm an advisor to the PETA Science Consortium International, and I'll be co-moderating the webinar today, along with Gino Scarano, who is a senior advisor in the New Chemicals Division of the EPA Office of Pollution Prevention and Toxics, as well as with Gavin Maxwell from Unilever, and Aaron Schlengster-Schmierer from the Physicians Committee for Responsible Medicine. Hello, everyone. So today's webinar is part two in a two-part series. Part one was held on December 7th of last year. And the slides and a recording from that part one webinar are available at either of the links that are on your screen now. And all of the materials on the use of the series on the use of NOMs and risk assessment, as well as a neurodynamic discussion series, are available on those websites. And the recording and presenter slides from today's webinar will also be available there as soon as we're done with the webinar. So today we have two speakers that will be presenting on frameworks for establishing scientific confidence in new approach methodologies. We will then go into a panel discussion with both the speakers as well as 3 additional panelists. Please use the Q&A feature at the bottom of your screen to enter questions for our speakers and panelists. You can enter those questions at any time. You're also welcome to upvote questions you like by clicking the little thumbs up icon. And then also, if you do submit a question, please identify if it's directed to a specific speaker or panelist, okay? I will now turn it over to Aaron to give us some updates. Aaron?
Thanks, Bridget. Thanks, Bridget. As Bridget mentioned, this webinar is a partnership between 2 ongoing webinar series. Because we realized we were trying to plan very similar events. So today's topic is on everybody's mind, so we hope you will enjoy and participate in the discussion. We're also hard at work planning additional webinars for the upcoming year. Head to pcrm.org/neuro for more information on our upcoming dynamic discussion ecotox webinar, as well as our upcoming carcinogenicity series. Please keep an eye out for announcements by joining both of our mailing lists, or by checking out the links Phoebe has dropped in the chat box below. Back to you, Bridget.
Thanks, Aaron. Okay, I'll now welcome Gino Scarano from the EPA to give us some introductory comments.
Thank you, Bridget, and hello, everyone. It's a pleasure to welcome the speakers, panelists, and all of you joining today to discuss part 2 of our webinar on frameworks for establishing confidence in new approach methods. As I said in my introductory remarks at part 1 on December 7th, the identification and use of NAMs by regulatory agencies in the US and around the world is a journey and a goal that has been going on for at least a decade. I'm a regulator and I'm very interested in this. I am looking forward to today's presenters, panelists, and all of you asking questions and providing comments on this topic as we all go on this journey together. So let's go, and thank you.
Great, thank you so much, Gino. So speaking first today is going to be Dr. Alistair Middleton. Oh. 1 second, y'all. Okay, so Dr. Middleton, he joined Unilever Safety and Environmental Assurance Center in 2014 after working in academia as a research associate in Germany and the UK, where he used mathematical models to understand the dynamics of complex biological systems, including gene regulatory networks and cell migration. He is now a science leader in computational toxicology, where he leads a multidisciplinary team looking at how to use computational models and in vitro data to make robust safety decisions without the use of animal testing. Thank you so much for being here, Dr. Middleton. Please go ahead.
Thank you very much. Just share my screen. Hopefully everyone can see my slides.
We have presenter view so far, or there you go, perfect.
Okay, great, thank you. Yes, so welcome everyone. Thank you very much for joining us. I'd just like to say thanks also to the organisers for giving me this opportunity to discuss some of the work that we've been doing around developing a toolbox and workflow for conducting systemic safety assessments without the use of animal data. Now, within that, it's not just about how you define what that collection of new approach methodologies is and how they work together, but also about establishing that scientific confidence that actually that overall approach is actually fit for purpose, and that you can use it to conduct those safety assessments so that you're arriving at decisions that are protective of human health. So the basis of this talk is this publication that was accepted to Toxicological Sciences earlier last year, and I provided a QR code here if people want to directly access it. Just to tell you a little bit about Unilever, where I work. We produce a range of different consumer products from foods, drinks, home care and cosmetics products. And to conduct those safety assessments, we use a risk-based approach, which involves looking at both the hazard and the exposure to evaluate the risk. And we can safely use, so that we ask the deceptively simple question of, can we safely use a certain percentage of an ingredient in a product? And that's to conduct the safety assessments, not just for the consumers, but also the workers and the environment. To do this, we have been investing heavily in building capability in next generation risk assessment, which is defined as an exposure-led and hypothesis-driven approach, which integrates new approach methodologies to assure animal testing, to assure safety assessments without animal testing. And this is an area that's been evolving over the last, at least the last decade, as was mentioned before, not least with the publication of toxicity testing in the 21st century, a vision and strategy, and has since been followed up by a number of different groups across the world, but not least the different federal agencies in the US, such as the US EPA, FDA, and also the National Toxicity Program. One of the most important aspects of next generation risk assessment is actually that the idea is to identify exposure levels that are protective of human health. And by no means are we attempting, for example, to predict toxicity endpoints, particularly in those sort of high dose animal studies that you might find. So the central concept is that if no if no bioactivity is observed at consumer relevant concentrations, there can be no adverse health effects. So you nicely see that illustrated in this diagram here, where you can see a comparison between typical exposure levels given by the green squares and then the points of departure at which concentrations that you might expect to see some bioactivity being triggered by the chemical. And in several cases, you see that there is what you might consider to be a margin of safety, or what we call a bioactivity exposure ratio, which basically reflects the fact that your exposure levels are way below the concentrations needed to trigger any effect. So NGRA uses new exposure science and understanding of human biology to basically conduct those safety assessments. Now, over the last few years, there's been a number of case studies looking at individual chemicals and exposure scenarios, trying to understand how we basically piece together the new approach methodologies within tiered risk assessments. So, for example, in our group, I'll just get the laser pointer ready. So in our group, we conducted one particular case study that was again published in Toxicological Sciences, which looked at coumarin in hypothetical consumer products. So I won't go into a lot of detail around this, but it was a basis of the work that I'll be talking about in the subsequent slides. So the idea is that we considered, how would we perform a safety assessment if we wanted to include 0.1% of coumarin in a face cream or a body lotion? And to do this, we adopted a tiered risk assessment format, where essentially the idea is that you're bringing together the different tools that you may have at your disposal, so the in silico predictions, in vitro cell assays, etc., to make that safety decision. So, for example, you would use information about consumer practices, or maybe in silico predictions of ADME parameters, to then use what are called physiologically based kinetic models to estimate internal exposure levels. That would be via this plasma Cmax, and then combine that with other in silico alerts around potential hazards associated with that chemical, and then bring that together with the in vitro cell assays, which are designed to really capture a broad range of potential effects that could be triggered by the chemical, which would be a toxicological concern. So those sort of assays I'll go into a bit more later on, but they were things like this cell stress panel, or looking at high throughput transcriptomics and so on. And then we bring those two quantities to essentially obtain from that points of departure, and bring those two quantities together in terms of this bioactivity exposure ratio, or as described here, margin of exposure, to help aid the safety assessor to ascertain the risk. Just want to point out also there's a QR code here again, if you want to read more about that publication. So that bioactivity exposure ratio is a key metric within the decision framework. It's not the only aspect of the decision-making process. We bring all the different levels of information together to make the final decision. So I just want to emphasize that, but it is an important number for conducting the safety assessments. So in this particular case, what we saw was, if we looked at the internal exposure estimates for the two products, we've got the concentration in micromolar here on the bottom horizontal line shaded region, and then we can compare that to the different points of departure obtained from the different technologies that we had. So looking at specific binding to specific targets here, and also looking at this cell stress panel, which measures effects such as oxidative stress changes, for example, and also high throughput transcriptomics. I'll talk a little bit about that later, but the idea is that essentially the bioactivity exposure ratio, so this difference between the two, or the ratio between the two, range between 158 and almost 100,000, so relatively large numbers. And this, together with the other sources of information within the study, suggested that the 0.1% coumarin inclusion in those two products was safe for the consumer. So this study really helped us and understand how we would conduct those safety assessments in general, but then this also led to a broader question over, what would be an appropriate toolbox of new approach methodologies for actually estimating that bioactivity exposure ratio that we could apply to a range of different chemicals and exposures. And so that's what we've been doing in the last couple of years. And so we've defined that for the moment as composed of this high throughput transcriptomics. So the idea behind that is to obtain a broad, untargeted measure of the potential effects caused by a chemical on the cell. And that's measured in several different cell types. this cell stress panel for measuring various, as the name suggests, cellular stress effects. And then also this in vitro pharmacological profiling, which is again for looking at specific targets of pharmacological or toxicological concern. And then combine this with the various models for estimating the exposure or analyzing the data to estimate these points of departure. But of course, within that, it's not just about defining that toolbox or the workflow, but also about building that scientific confidence that it's actually fit for purpose and robust. So there are a number of challenges that are very much part of that sort of question. One of those is really around the biological coverage. So the idea is that we've got a broad range of different cellular assays that cover a range of different potential effects that could be triggered by the chemical. So are we actually, do we have all the right cell models to have enough breadth in our sort of ensemble of different models to capture all those effects? Equally, are we measuring the right biomarkers? So that's something that's also, is high throughput transcriptomics, for example, sufficient, or should we also be looking at phenotypic profiling? And then there's also, on the other side, on the exposure side, the relative accuracy of the terminal exposure estimates provided by the PBK models. And we know that this can vary according to how those models are constructed and parameterized. And I'll talk a little bit more about that later on. And then there's this question of how large should the bioactivity be, but this bioactivity exposure ratio be, sorry, to identify something as low risk. So it's very much about trying to evaluate that toolbox and see if it's really working well so that we can make those safety decisions with confidence. And so to do this, what we did was we took a benchmarking-based approach. So we basically, the idea is that you identify both low-risk chemical exposure scenarios where there's already existing evidence to suggest that basically they are low risk, there's no cause for concern, and they're typically ingredients to foods and cosmetics. And then you've got the high risk chemical exposure scenarios, which again, there's evidence that they cause maybe some systemic side effects, perhaps relatively mild, but nonetheless things that you don't want from a consumer goods perspective, or things that really cause adverse side effects. And then once we've identified those chemicals and exposure scenarios with which to basically measure the overall performance of the toolbox, we then treat them as though they're basically part of an ab initio risk assessment. So we pretend that we don't know anything about them, and we apply the tools as though we were conducting a normal NGRA study using this set of tools. So in other words, we would run the PBK models to estimate the exposures. We then run the in vitro cell assays to estimate those points of departure, and then calculate the bioactivity exposure ratios and see basically what happens with the data. So if the approach works well, what you would expect is that the bioactivity exposure ratios for the high risk would be relatively small, so typically below 1, reflecting the bioactivity that those substances are thought to cause in humans, whereas the low risk bioactivity exposure ratios would have, so the low risk exposures would have high bioactivity exposure ratios. So in other words, you could use this quantity, this BER quantity, to really distinguish between those two groups. But also you could also start thinking about what the threshold is at which you can start saying something is low risk. So what we've done is we've broken this up into 2 stages. So the first step is to define pilot study, which is basically the basis of this publication that I mentioned before. So that's where we define exactly what the toolbox is, what the workflow is, how the toolbox should be evaluated, and what the metrics should be to measure its performance. And then we walked through that essentially with a small set of chemicals and exposure scenarios, which I'll go into further details in the next few slides. And then we've also proposed a sort of prototype decision model on determining that threshold BER, so that when you do get a new chemical, a new exposure scenario, you can say its relative risks based on that BER threshold with some confidence. But then, of course, that's just the overall design of the study. And then we are in the process of then performing this, what we call the full evaluation, where we then really test the toolbox with a wider set of chemicals, so roughly 40 chemicals, and then I think it's almost 100 exposure scenarios. And then we're going to use that to basically, look at how well the toolbox performs, and maybe use that to potentially even refine the toolbox in terms of its composition and that decision model. So this is so just to walk you through that pilot study, the results that we got. So these are the sort of sets of chemicals that we identified. So you'll see that for some substances like caffeine, that I'm sure we're all familiar with, you've got actually three exposure scenarios, two of which are low risk, one of which is high risk. The two low risk ones are, where it's included in a shampoo or of course, as a component of coffee. But then also the high risk one is related to poisoning, where too much caffeine was essentially ingested. And then for each of those chemicals, we then generate these points of departure. The only thing to really add to this diagram that I haven't mentioned before is really that what we do is when we've looked across all those technologies, you get a POD for each. And then we take essentially the minimum point of departure to estimate the bioactivity exposure ratio, since it represents the most conservative estimate of the potency of the chemical. So this is an example of some of the concentration response data we get. So at the top here, we've got high throughput transcriptomics data. At the bottom, it's the cell stress panel data. So again, you do the curve fitting and you estimate the PODs. And then this is a summary of all the different points of departure we get across those different chemicals, really showing the sort of range of potencies we have from niacinamide at the top and caffeine, all the way down to doxorubicin, which is a chemotherapy drug. But also what you can see is that depending on what chemical you're looking at, the key sort of the key points of departure, which would drive really the risk assessment, depends, can vary in terms of whether it's coming from this in vitro pharmacological profiling or one of the cell lines we used in the high throughput transcriptomics, or potentially the cell stress panel. So that's really underlying this issue around that biological coverage. Then we move on to the exposure estimation, where again we use these physiologically based kinetic models. But the important thing here is that we really approach it in a way that it's reflective of the true risk assessment process, where you've got in silico. So the idea would be that maybe at that first tier, you would maybe only have in silico parameter estimates for those PBK models, or maybe further down at the next tier, you may have generated some data for in vitro parameter estimates, or perhaps even with the risk assessment being even more further progressed, at the highest tier, you may also actually have generated some human PK data. So basically, what we do is we generate PBK models for each of those conditions and obtain the plasma Cmax estimate. And what we know already is that basically, if you take a model that's based purely on in silico parameters, it will be far less accurate than a model where, for example, you've been able to use some clinical data to help parameterize the model. So what we find is, so what we did was we to study that further, we took a series of chemicals and exposure scenarios where we already had measured Cmax. And then we ran the models for each of those cases and then basically estimated the relative error we got. So what we found was, again, with the in silico parameter-based models, we got errors that ranged between 10 and 100, say, when you had some in vitro parameter data. So, for example, we were being able to get a better estimate of the clearance rate in the models through that, then maybe the error varied by tenfold. And then maybe in the case where you had this clinical data, you would get down to sort of more 2-3-fold differences. So that's all very well, but we also generated developed a Bayesian statistical model that allowed us to actually quantify the error, so that based on that past performance that we know for other chemicals, we could then say, okay, given a new chemical, this is the kind of error we could expect, but do that in a probabilistic way. We then brought those two sort of quantities together, as I mentioned, but the important thing is that we use that Bayesian statistical model to actually provide a distribution for what we think the true BER is. So that distribution basically reflects that uncertainty we have in the PBK model estimates. So what we have here is a list of chemicals and exposure scenarios. So again, that we used for the benchmarking, again, color-coded against the risk categories. And what you see depicted is the 95% credible ranges of those distributions. So what you can see is very nicely, much like that cartoon I showed you earlier, that the bioactivity exposure ratios for those high-risk substances are generally below 1. But as you walk down the list, the BERs get larger and pass above 1, and then particularly for those low-risk chemical exposure scenarios. But not only that, you can use these distributions to say, for example, here you've got a relatively narrow range for the doxorubicin, and so you can say there's a very high probability that true BER, that value is definitely below 1, right? So you've got very high confidence of that. Even given that uncertainty in the accuracy of the PBK model. Whereas if you take, for example, the hexaresorcinol, what you can see is that distribution really straddles the BER equals 1 line. So there's, given the uncertainty in the accuracy of the PBK model, you can see it's not quite clear whether or not the BER is above or below 1. And that very much makes sense given what it's for, because it's for hexaresorcinol, which is an active ingredient used in throat lozenges, for example. And then, as you go further down, you've got here you've got coumarin, again used in food products, where you can see that that distribution is way above that BER1 line. And so you can say, okay, there's a very high probability, again, that this Cmax, the Cmax estimates are really way below the points of departure. So you've got high confidence of that. But then there's a question of how you use this probabilistic information within that decision model that we want to come up with. So just to cut a long story short, what we did was we said, okay, we want to assign a probability target. So we said, okay, we want to say, okay, there has to be a 97% probability, for example, that the true BER threshold value is above 1. And as long as we're above this, we can call something low risk. So this is essentially what we did. So for example, for level 2, so this is where PBK models were based on this, where you've had in vitro data. Any BER value taken from your PBK models and your points of departure, provided that value was above approximately 10, then we would call that a low risk chemical exposure scenario. Anything below that, we would call uncertain risk. And the point here is that what we want to see was actually how well did that work when we compared to the benchmark data. So how well did the toolbox actually perform in reality? And what we see is when we use that decision model together with the data, we're able to support some of those low-risk chemical exposure scenarios while rejecting all the high-risk ones. So in other words, we're 100% protective against those. But of course, that's just the beginning. As I said, that's about designing the sort of framework for establishing that scientific confidence that it works. But now we're in the process of generating data across 40 chemicals and almost 100 exposure scenarios. And really the idea is to run the data through those exactly as I've laid out here, but then really see how well the toolbox performs, and really to take an iterative approach to this. So then, you know, I don't doubt that there is going to be shortcomings in the toolbox that will be highlighted through that testing. But the idea is to then evolve it further, maybe by changing the composition or improving the decision models that we're using, so that we can improve it and make better safety decisions with it. One thing I would like to point out is that we're very interested in how this sort of idea around how we establish scientific confidence aligns with, for example, the framework that was laid out by the Van der Zalm paper that Dr. Kleinstra was talking about in the last session. So that's something for us to be working on this year. And then another thing I'd just like to stress is that we're working with the US EPA on a number of things, but not least generating more data for these chemicals in the full evaluation across more cell lines using high throughput transcriptomics and phenotypic profiling, so we can get a better understanding of that biological coverage question. So again, hopefully one day be able to tell you more about that in the future. Thank you very much for listening to the talk. I'd just like to thank my co-authors and also collaborators at the US EPA. And if you want to learn more, please, this website hosted by Unilever will tell you more about the work that we're doing. Thank you very much.
Great. Thank you so much, Dr. Middleton. I see we're already getting some questions in. As a reminder, we're going to hold all questions until the end of the next presentation, but do feel free to keep adding more, and just please point out if it's for a specific speaker, okay? All right, speaking next will be Amanda Ulrey. Ms. Ulrey is the Vice President of Business Operations for the Institute for In Vitro Sciences, or IVS. She provides leadership, training, and oversight of the Client Services, IT, and Quality Assurance Unit, and institutes and maintains policies and procedures for regulatory compliance and organizational quality management. Ms. Ulrey is responsible for the development and launch of IIVS's business-to-business certification in Good In Vitro Method Practices, or GIVIMP. She has been a registered quality assurance professional in good laboratory practices since 2005 and has presented, trained, and authored white papers on the topic of the application of GLPs to in vitro studies, the OECD GIVIMP guidance document, the importance of supplier qualification programs, data integrity, and IT compliance and computer software validation. Welcome, Ms. Ulrey, please go ahead.
Thank you very much, Bridget. Can I just get a confirmation that my slides are visible quickly?
Yes,
Looks good.
Fantastic. Okay, thanks everyone for tuning in today. I'm going to be speaking about how following the Good In Vitro Method Practices aligns your work with the framework. So the first question is probably, what framework am I talking about? So that goes back to part 1 of the webinar series. I'm specifically mapping out GIVIMP against the points that were brought up within the Van der Zalm paper, a framework for establishing scientific confidence in new approach methodologies. So if you haven't watched part 1 of the series, I would highly recommend that you go ahead and go back and do that at some point. That will give a lot more detail and specifics to what I'm going to be talking about today and put my thoughts into more context. But just as a very quick refresh, that paper builds on information compiled over several years. You can take a look at the list of authors. They've been within the NAMS community for a long time, so it's an excellent compilation of best practices for building scientific confidence in NAMS. And although the paper speaks of the original focus being on pesticides and industrial chemicals, as it was pointed out in part 1 of this webinar series as well, the applicability of the concepts that are raised within that document is much wider than just pesticides and industrial chemicals. So it's a very broad paper. And again, as a refresher, The framework spoke about 5 different elements for establishing scientific confidence in NAMs. The showing fitness for purpose, human biological relevance, technical characterization, data integrity and transparency, and independent review. And all of those things can be performed at different times. There's not a specific order. So that's a brief refresh on the framework. So GIVIMP, for those who might not already be familiar, is a document that was published by the OECD in 2018 on good in vitro method practices. It is a detailed and very widely applicable document for laboratories. So it has a lot of information for what you can do within a laboratory to follow the best practices that are state of the art for in vitro work. It gives you guidelines and thoughts that are aimed at improving the test methods. So it's not simply a compliance guide like good laboratory practices might be. It touches on elements of scientific design and technical characterization that are helpful for in vitro methods, specifically things like discussing your applicability domain, any kind of assay limitations, and those topics aren't generally covered in other quality type guidelines. So it's very helpful that that's covered within GIVIMP, because then following GIVIMP can give labs a blueprint for building a thorough paper trail so that they have all the evidence they need and can provide explanations further down the line for all of these items. GIVIMP also covers aspects of the work that impact the reliability and relevance of the in vitro data. So reliability and relevance are really key in supporting scientific confidence in a method. And lastly, again, GIVIMP provides a structure and a way to move forward so that as you're developing and performing in vitro methods over time in your laboratory, you build things like acceptance criteria and performance standards into your method, which improve transferability and repeatability over time. So we have the GIVIMP document and the framework document, and they're both really focused on improving the confidence ultimately. The framework specifically discusses how to build scientific confidence as you're looking at a number of different methods. You can assess it against the framework to see if you feel that there is enough information for you to be confident in the new method. And GIVIMP gives you information on how to structure and manage your laboratory work and what actions you can take during and throughout method development to assure that you'll be aligned with the recommendations of the framework at the end. So following GIVIMP should affect how you are performing your work in the lab and provide some helpful changes maybe to your actions within the lab so that moving forward over time, you're moving forward to build that scientifically relevant and really robust data sets and methods. And then looking back again, once you're at the other end, when the lab work is done, you can say, yes, I really do have confidence in these methods. We're aligned with all of the recommendations within the framework. So going back to the 5 points within the framework, there are 3 that I think GIVIMP really provides some specific guidance for. The technical characterization, data integrity and transparency, and recommendations around the independent review. For technical characterization, you can look within GIVIMP in Chapter 4, which discusses the equipment that you're using, apparatuses, materials that are used, reagents. Also Chapter 6, which discusses test and reference control items. Chapter 7 on standard operating procedures, and Chapter 8 on overall performance considerations for the method. For some detailed guidance on data integrity and transparency, you can look in GIVIMP Chapter 9 on reporting the results and Chapter 10 on storage and retention of records and materials, which is where they really have a whole section on data integrity. And then all chapters within GIVIMP, since it's designed to help improve reliability and relevance of methods, make sure that any independent review, whether that's a validation authority, peer review, a editorial review prior to publication, that you would have established all of the information that would be required or asked for at that point. So looking more detailed at technical characterization, the column on the left there, I pulled out some points within the technical characterization section of the framework. And then the column on the right gives the mapped sections within the GIVIMP guidance document that have specific actions to take within the laboratory. So, for example, in the framework document, they made a point to assess and describe the methods. And in GIVIMP, Chapter 7.1 on standard operating procedures development really goes through in detail the process for developing an SOP, what the stages might look like, because you're not going to obviously start with a fully developed and realized SOP for explaining the method. So Section 7.1 kind of walks you through that, which is a whole process for describing your method. And same with Chapter 8, Section 2, discusses some elements of experimental design that should be considered when you're describing your method, specifically some guidance on plate layouts, solubility. So really some detailed points to consider that would be helpful as you're moving through trying to get some technical characterization and really nail down the specifics for a new method. The framework discusses transferability of the method. In GIVIMP, Chapter 8, Section 4, talks about proficiency chemicals and the importance of selecting proficiency chemicals and using those to show that a method has been properly transferred to new laboratories. Applicability domain is an important concept within the framework document, and there's an entire section within GIVIMP that talks about how you describe and define the applicability of your method, including any limitations of the method. For reference and controls, there's an entire chapter on reference and controls in GIVIMP with points to consider there. And the framework also brings up the need to define and describe limits of detection and quantification. And within GIVIMP, you can find some guidance there in Chapter 4, which discusses some of the apparatus that might be used in gathering data, also detection limits and cutoff values, and linearity and dynamic range of the apparatuses that's discussed in Chapter 8. As well as accuracy and precision. That's also recommended in the framework and discussed in Chapter 8 of GIVIMP. So following GIVIMP recommendations while you're performing the work really sets the method up to be able to be evaluated as recommended by the framework document. So the framework says to evaluate the protocol, the equipment used, and any computational models being used for endpoint prediction. So evaluate the protocol, and GIVIMP gives you some information on how you can evaluate the protocol and what you can evaluate it against. So you can go through GIVIMP and look for what the recommendations are depending on the type of method you have. If it's a monolayer cell culture-based method, if it's a three-dimensional tissue-based method, there are recommendations for all different types and for what should be considered and defined and described within the protocol. So another item for technical characterization or another framework that you can reference is this paper by Peterson et al., the technical framework for establishing high quality measurements in NAMs. So this paper, this framework can be referenced through I would say through the method optimization stage, because high quality measurements, endpoints to consider are part of the characterization of the method, but you need to start nailing down some more of the specifics of your method before you start getting into mapping out cause and effect diagrams, for example. And when you do this work, you end up with a robustly defined method at the end of it. So all of this feeds into your method design and your general protocol, study plan, SOP specifics. So I would also recommend taking a look at this paper and this framework when you're looking to optimize any new methods that you have. For applicability and limitations of the method that are called for by the framework, GIVIMP also discusses the need for transparent discussion and reporting of the applicability domain and limitations of the method. And when you go back and look at part 1 of this webinar series, there was a lot of discussion at the end about how we don't want to move a method forward and just say what it is for. We want to be able to say what it could be for so that you can then apply it potentially more broadly and build on the applicability domain. And in order to do that, you can apply the work and the framework and the method in different regulatory frameworks if you have a well-defined applicability and limitation. Same with different chemistries of the test items. You can start in one place and have the information there from your starting point, and then moving forward, you can assess against different chemistries and types of test items and see if there needs to be additional work done to expand the applicability domain, or if there are pre-existing limitations of the method where you should look for something else in that context. So having that as part of the process of defining and characterizing your method makes it easier down the line to continue to use it in new contexts where appropriate. And reproducibility and transferability, the framework defines reproducibility as the measurement of how closely results of a particular method agree when repeated using the same protocol and substance. So if you're not documenting what you plan to do, or at the very least what you did, and carrying that information forward, it's incredibly hard to reproduce the work that you're doing. So this always made me think of the ESTIV meeting presentations that were focused on having well-documented and defined materials and methods in manuscripts. This is the same thing. When you have a robust method and you have it defined and described to an appropriate level of detail, the method is more reproducible by others because they have your instructions, and it's easier to transfer to new laboratories. Following GIVIMP leads to the development of these strong materials and methods sections so that it can move forward and be more easily reproduced because it is more specific. It also has some really specific recommendations concerning training and transferability and the use of proficiency chemicals that also help with training or with transferability. For data integrity and transparency, the framework requests that you demonstrate the integrity and credibility of your data, from raw data to the final report. Within GIVIMP, in Chapter 10, they discuss ALCOA, which anyone who's familiar with a regulatory context and good laboratory practices should be familiar with ALCOA. That is an acronym used to describe the attributes of data and data integrity. So data must be attributable, legible, contemporaneous, original, and accurate. So that level of detail, which is important if you're in the laboratory because it starts at data capture, you can get that detail from GIVIMP and Chapter 10 specifically. And then independent review that's discussed in the framework suggests demonstrating data integrity prior to the independent review process, and in addition to the scientific review, having an evaluation of the process used for data acquisition, transfer, and processing. So if you haven't been building and maintaining your data quality through good data governance throughout the process of doing the laboratory work, it will be difficult to show this part of what is discussed within the framework at the end, because you can't insert it later. So following GIVIMP, if you're doing the recommendations and the points to consider within that document while you're doing the work in the lab throughout your data collection and analysis, then you will have what's needed to show data integrity and to have a full, robust body of work to show any reviewers at the end. So in summary, I think it's important to think about the concept that the quality of the data can't be improved once it's collected. The data are the data when you collect them. They don't get better later. But data can lose integrity unless you're handling it properly throughout the process. So there's no amount of assessment that you can do at the end of your laboratory work that could go back and fix the quality of what was done before. GIVIMP gives you best practices to follow in the lab for your new method design, execution, and reporting. So through the whole process, it gives you your steps so that you are collecting strong quality data and you're maintaining it in a way so that it will all be there when you get to the point of review. So GIVIMP gives you your laboratory behaviors to follow so you can meet the framework and improve overall scientific confidence in NAMs. One thing that is brought up within the framework paper that I disagree with slightly is at the end of the paper, they discuss assessing against GLP-like frameworks. And I think that can be interpreted many different ways, the term GLP-like. And I know from personal experience, it's difficult to bring essentially a regulatory concept into a space where it's more R&D and you're doing more research and development work, and it's not part of the regulatory framework. And I don't think you need GLPs or GLP-like guidances because GIVIMP exists, which was written specifically for in vitro method design and performance. So that's the end of my talk. Feel free to send some questions. I have a QR code here in case you need or would like a copy of GIVIMP. You can scan that, it'll take you right to it. And thank you very much.
Thanks, Amanda, for that important presentation. We will now move on to the panel discussion. we have 3 additional panelists joining our speakers today. First is Dr. Anna Lowit, who is the Senior Science Advisor in the Office of Pollution Prevention and Toxics. She advises senior managers and leads multidisciplinary teams on a variety of cross-cutting topics related to human health and ecological risk assessment. She is currently one of the co-chairs of the Interagency Coordinating Committee on the Validation of Alternative Methods, or as you may know it as ICVIMP. Next, Dr. Helena Hogberg-Durdock, who joined the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICEATM, within the Translational Toxicology Division at the National Institute of Environmental Health Sciences in December 2021. She is conducting research drawing on her broad expertise in applying in vitro methods to assess the developmental neurotoxicity potential of chemicals. Previously, she was the Deputy Director of the Center for Alternatives to Animal Testing within the Johns Hopkins University Bloomberg School for Public Health. Let's also welcome Dr. Susanne Kolle, who currently heads BASF's SE's Laboratory for Biokinetics and Local Toxicity and supervises the field of local toxicity, including eye irritation and corrosion, skin irritation and corrosion, and skin sensitization. Susanne has led the implementation of good in vitro method practices at BASF. She is also a member of the scientific expert groups in the field of local toxicity and has authored numerous publications. Thank you all for joining in the discussion. I see a ton of questions flowing in the Q&A, so make sure you thumbs up the questions that you want answered so that we make sure that we get to them and also keep them coming. Dr. Gavin Maxwell is here helping me with the Q&A session today. Gavin, do you happen to have a first question for our panelists?
Yes, thank you, Erin. I'm going to go with the top voted question at the moment, which is one for you, Alistair, but one the rest of our panel may also have a view on, which is when you use in silico data for regulatory decision-making, do regulators tend to scrutinize the modeling platform on a deep level, for example, reviewing code, understanding training sets, etc. And does the scrutiny decrease with experience reviewing data from the same platform, or does every application need the same vetting? Alistair, do you want to start by answering that, and then I'll put it to the wider panel if they've had any experience on this?
Yeah,
question.
So I mean, as I understand it, I think that very much depends on the regulators. I think some regulators would be interested in looking at the code and understanding the models at a deeper level, whereas in other cases you may have some sort of software tools that are generally accepted, and therefore, you know, they would just wouldn't necessarily look at it at that level of depth. And I think just to say also, I think in the area that we work in, we're developing a lot of novel tools, and therefore there's a great deal of sort of challenge around that area actually in terms of the regulatory acceptance of them, particularly in some of the work I was talking about, for example. But sorry, yeah, probably other panel members have maybe more other things to say on this.
Thanks, Alistair. Does anyone else have a view on this? Maybe what more we could do in this space to establish standards for reviewing these kind of data or platforms? Anna, maybe?
Yeah, thanks, Gavin. And first, this is my first PUP webinar, and I was just amazed at the international participation and the numbers, and I'm really enjoying the presentations this morning. So just wanted to say thanks for everyone for being on, and including me. It's fun to do these kinds of things, and it's fun to listen to other people and learn some things new, and to see that really the practical application and testing out of the framework that a number of us put out last year. It's just really fun and cool to see. So I think I'm the sole regulator on the panel, so I think I'll take a stab at this one. I think what Alistair said is true. I think it depends on the country, on the regulatory authority, and on the situation that, you know, like if you look at the US situation, depending on which agency you go to, whether it's EPA or even other parts of different parts of EPA or FDA or other places, each organization sort of has its own way of looking at things. And so we always encourage whether it's model developers or in vitro method developers, to really engage with the target audience for their work. And in fact, under the ICFM strategic roadmap, one of the things we really talk about is the need for method developers to engage directly with end users, and that connection and that interaction is really vital to the development of methods that are relevant for regulatory use. But for the most part, as a regulator, I do have an expectation that the code is accessible, preferably publicly accessible, but at least accessible for external peer review. I will tell you that just a publication is not enough. Just handing me a publication, although that's a wonderful achievement, is not enough from a regulator to reach to the level of peer review and expectation for use. And what that means really varies on the situation. So it's better to really talk to the regulatory organization you're targeting. But a publication a single publication is just not ever going to be enough. So I'll stop there.
Thanks, Hannah. Brilliant. Anyone else want to come in on this one, or will you move to the next question? I'm not seeing anyone on mute, so I'm going to hand it back to you, Erin, if there's another question.
Sure. I have one. Is there a panel of in vitro in silico tests for general toxicity that you recommend for NAMs that have not been tested? Anyone have any feedback on that? Okay, I picked a bad question.
Yeah, no, so I mean, I can say, something. I'm not aware of one, to be honest with you. But what I would say is, I guess what we're potentially looking at is, to what with the toolbox that I described, for example, we're looking to apply it within the space of systemic toxicity, but then also it could be useful in other areas as well. So that's, I would say, I don't know about general toxicity, but certainly understanding how these different NAMs can be used in different contexts is really an area of active research and development. Sorry, I don't know if anybody else has anything to add to that.
Yes, I think the question is really broad, and general toxicity is really broad. So there's no one size fits all toxicity test or panel off. So, and I guess in a world where we're moving towards more mechanistic information and pay particular attention to biological relevance and things like that, there won't be one assay that fits all purposes.
Okay. Gavin, do you have another question?
Yeah, I'll go with the next one that's got the highest votes, because I think it maybe does build on this. So, Alistair, I think it was particularly motivated by your talk, but I'm sure others will have a view as well. So what percentage of the toxicology data you use for the development of the toolbox is derived from animal studies, and how much from human studies, and how to avoid any bias that may be there due to the animal data?
Yeah, so the only way in which the sort of it's more indirectly the animal data only plays a role in the sense that it has been used in the past to conduct those risk assessments and to determine the safety of those benchmark substances. But then of course within that you've also got safety decisions that have been made through other as products have gone out and then be recalled or whatever. So you'll have also human data within that as well, or revisions based on observations or other measurements that have been taken since those initial assessments. So to be honest with you, I don't know what percentage. None in a direct sense, but of course we're building on past safety decisions, essentially. Yeah.
Thanks, Alistair. Does anyone else have a view on this one? I think this is quite an interesting question in terms of how do we build on our experience with animal data, but also maybe where are there opportunities to use more human data when evaluating NAMs or IATA?
Well, I will take a shot on this.
Thanks,
I don't
Helena.
know the percentage, but I mean, there are certain endpoints, for example, the skin sensitization, where we have a lot of human data, which is, you know, superior to the animal data. I'm also going to do a parallel to developmental neurotoxicity, where we actually do not have any or not a lot of animal data. So there we are taking an approach where we're trying to develop the NAMs for human relevance. And that is, you know, where we're trying to go, because there's just no animal data and there's no way we will generate that data either, because it's too complex and too difficult. Then I will also, I'm sure that like Nicole talked about that on the last time, but we are updating the validation document, the ICFAN validation document, that's hopefully going to be available in the spring sometime, and or at least next year. This year. And there we are talking a lot about human biology and how, you know, you need to, if there is human biology relevant or that is available, we should rather compare to that data than to the animal. But of course, different regulators have different opinions in this as well. So I think we'll see how it goes. And we're trying to work a little bit with pharma currently to try to also take in some more data that might not be available to us normally, but try to link to that kind of information as well.
Thanks, Helena.
Maybe to add on this, I think it's phrased correct in the question, avoiding the bias towards animal data, because what we have been using in particular in regulatory toxicology in the past is animal models, and we all know they're only models, and they have their mistakes and variability, and also, so I don't want to go into details here, but I think, yeah, time is right to change mindsets here, and really not trying to replace one model by another model, but really go towards the biology and human-based toxicology.
Thanks, Susanne. And Anna?
Yeah, I think yeah, I agree with Susanne, what Susanne just said. And there's, you know, there's a publication that a few of us put out a couple of years ago on basically testing out this idea that using human biology to look at the human relevance of the models, whether it's the in vivo or the in vitro. So there's a publication, Amy Klippinger is the first author on the eye irritation. And we're in the middle, we're pretty close to submitting a similar parallel publication on skin irritation, looking at the animal and the in vitro models to sort of explicitly look at human biology and the relevance and the appropriateness of the animal models and the in vitro models to sort of advocate, you know, for whether, you know, how to choose which way to go. And people are thinking about sort of starting to really actively do this. And I think one of the challenges that we have, the DNT is a really good example that, you know, the DNT in vivo study, first, there actually aren't that many. They're probably only 100, 150 ever done anywhere. And they're pretty much all in the pesticide space. And, you know, those studies are notoriously difficult and hard to interpret because the error bars on most of those measurements are extremely high. And I don't know of any that have been duplicated because they're so big and expensive. And so using that human biology as the primary anchor to validate and move forward is really the direction we have to go. And I think as we move towards thinking about cancer bioassays and things like that, we have the same issue that, you know, what's the relevance of the animal model in the prediction of the human outcome, and how can we get back to human biology and really anchor on human biology and linking that human biology to adverse outcome pathways, and how we then make that connection to the in vitro studies is really the direction our community is headed.
Thank you, Anna. Brilliant. Anyone else? I think we're done on that one. Erin, back to you.
Sure. We'll go to the next highest rated question. How do you know that the in vitro bioactivity panel you used cover the full toxicological space, including carcinogenicity, reprotox?
Yeah, so sorry, maybe there was a misunderstanding there. So we're not looking to never necessarily cover all toxicology in that sense. It was only for systemic toxicity, so it's covering, various organ toxicities. In terms of, for example, developmental, actually, that's something we're looking at in a separate study where we would the idea is that we'd essentially add on to the toolbox with specific assays for reprotoxicity. So, and also have to say that we don't know at the moment. The point is that we've essentially developed an approach for how we would evaluate it, and that actually we are undergoing that evaluation. But I think the point is, so how do you actually evaluate these tools and approaches to cover off the issues that were highlighted in the question, and then basically generating the data to assure ourselves that is the case, and evolving the toolboxes. So it's very much a journey and a learning process as well as, so it's not that we're saying, okay, this is it, this is definitely going to work and we're going to prove it. It's more, we're going to conduct these evaluations, see how we go, publish them, and then evolve the toolboxes as need be, depending on how those evaluations go.
Anything else, or move on to the next question?
Okay, I'm going to jump a bit further down for this one, so that because I'm feeling we're not testing Amanda hard enough here. So here's a nice one from Annie. Should there be another A in ALCOA, i.e. accessible? These in vitro data need to be discoverable in systematic reviews for risk assessment. What do you think about that, Amanda?
Yeah, I think that's a great point. And actually, the current acronym for ALCOA actually is called ALCOA Plus. So that's what we're doing these days. The plus is inclusive of consistent, complete, and enduring. So that still doesn't cover accessible. I think one of the points in the accessibility kind of goes back to having well-defined materials and methods in your publications, and that maybe it's not specifically a data integrity concern, but it is a concern for the reproducibility of NAMs as a whole. And I think certainly following forward with GIVIMP and any kind of looking backwards with the recommendations of the framework, once you do all that, you do need to make it accessible. So I don't know if anybody has any further thoughts on strategies for how to do that, or we could certainly look for it when we're doing review of manuscripts.
Thanks, Amanda. Yeah, have anyone got some other views on that? I think having a increase in the amount of in vitro data and databases is obviously something that's happening quite a lot. And obviously one of the important aspects of that is data quality and making sure these are data that people can trust and use when they're making risk decisions.
So again, I want to highlight a project that I'm involved in, which is called Methods to AOP, where it's a very broad international group. It started from the GRC with the idea where we are trying to link the methods in the adverse outcome pathways and have them quite well described. And we will come out with some guidance information to the OECD on what kind of information needs to be included into those things. Because I have to say also, it's very frustrating when you go, both if you use the adverse outcome pathways, which is maybe okay if it's endorsed, but there's a lot of adverse outcome pathways where actually there's no methods there at all. And each key event should be measurable. So there should be some kind of information about that. We are discussing currently how much information needs to be provided, because obviously it's not the methods developers that are developing the adverse outcome pathways necessarily, but we're trying to get the communication between those and try to promote also developers to provide this kind of information, which I think is really key. The same for publications, because exactly what we just heard, it's very difficult to reproduce because there's not just enough information in the publications. So yes, it's going to be important to share SOPs and detailed information going forward, I think.
Thank you, Helena. Any other views on that one, or we move on? Nope, over to you, Erin.
Cool. I think we kind of touched on this already, but just to sort of lay it out there, what is the regulatory acceptance appetite for NAMs as superior to animal models? I ask because liver toxicity via NAMs is empirically proven as superior, but animal models still seem to be de facto gold standard. Any takes on that? What is the regulatory appetite for NAMs as superior?
All right, well, I'll start as the only regulator, and then someone else can. Everyone's waiting for me, I think, to speak. So, you know, from a regulatory point of view, it's difficult to just pluck out a hazard that's unique and only test that, because if you go back to what the in vivo guidelines look at are multiple organ systems, so multiple adverse outcomes, whether it's the short-term or the long-term studies. So although I would agree with you, the liver assays are extremely advanced, just looking at the liver doesn't solve, from a regulatory point of view, other health outcomes and other potential hazards. So what we need to be doing as a community is developing and bringing forward more assays for other organ systems or other pathways or other tissue types that are equally ready for prime time. And I think the other challenge there that no one's mentioned that's really important in this space is that regulatory acceptance with respect to internationally harmonized, agreed upon protocols. And that although some organizations, including EPA, can and do accept non-guideline assays, our regulatory partners in other parts of the world and some countries cannot do that. And it really it is important that the OECD start to move faster to accept, to develop new guidelines and to move faster and with respect to developing internationally harmonized protocols. Because even if EPA accepts something, if our European or our colleagues in various Asian countries don't do that, we're actually not replacing anything and we're not making progress. So it's a very complex problem. And although you're right about the liver assays, it's just a piece of a very complex jigsaw puzzle.
Thanks, Anna. Does anybody want to add to that, or should we move on? Okay. Gavin, do you have another one?
Yeah, I've got another one. I think this might have been motivated by Alistair's talk, but there's probably something in this to consider for all of us. But do any of the exposure scenarios that have been used to develop and evaluate the toolbox consider mixtures of chemicals that cause similar responses or has similar mechanisms of action?
So specifically about, do we consider mixtures essentially?
Yeah, and I think whether it's an additive or a synergistic effect that we can pull out.
So in this work, no, we haven't looked at mixtures, but that is again something that is of course very important, well, not least to us as a company. So we as far as I know, we are looking at that in terms of how we model those mixtures, and also There has been some discussions on how we then integrate that within the risk assessment framework. But again, that's very much a work in progress, I would say. But it's definitely an important area to be looking at. Yeah, I don't know if others have anything to add to that.
Thanks, Alistair. Has anyone else got any experience on best practice when tackling mixtures with NAMs?
No, Gavin, but I think it's worth saying that for in vivo, we still don't have good best practices either. You know, and so I think it's one of those situations. Let's not hold the NAMs to a standard we don't hold the in vivo studies to. And in fact, I think one of the ultimate promises of NAMs is actually the ability to do more mixtures than we can in vivo studies. And so, you know, I think there's a lot of interest internationally in developing that science, but, you know, there's some steps to get there. You know, a number of people have tried in some assays to look at mixtures and formulations. And I think in my view, the success is a bit mixed and depends on the constituents of those mixtures and formulations, because something that has a lot of surfactants, for example, it's really hard to test in vitro because all you're doing is busting the cells open no matter what kind of cells you're using. So it's a complex problem, but it's one that we can solve with in vitro that we really can't solve in vivo.
Thanks, Hannah. That's a really good reflection on that one. It gives me hope. Any other views on that one, or we move on to the next question? I think over to you, Erin.
Sure. I think this is an important and a great question. I'm not sure we have a super clear answer, but let's see what you guys think. How can we stretch an in vitro result to a full physiological system, particularly for a more complex endpoint? How much different assay data would be enough for a regulatory decision on safety for humans? We might do many assays, but not address the right endpoint. Any reflection on extending in vitro, really making sure we make use of our in vitro data, the most use?
Well, I kind of like the IATA approach where you can do your in vitro, you know, screening approach, and then based on, you do a weight of evidence, and then based on if you think it's enough or not, you can go further. And there, I mean, there are some more physiological relevant in vitro models out there, microphysiological systems, combined microphysiological system, for example, and that could be the next step if that's what you feel is needed as a regulator, I guess, when you're supposed to take the decision. I mean, and then if that's not enough, then you might have to consider an in vivo, and depending on what you're looking at, what species, and could be enough maybe with a non-mammalian species, depending on what information you have earlier on. So I think this tiered approach, I mean, if you feel that you have enough information at an early stage, you can make a decision. Like, you know, for example, we have, again, I go back to the DNT because it's my background, but there we have an in vitro testing battery that the EPA actually used that to waive an in vivo study because they felt they had already some in vivo data on another chemical, and they saw similar response in the in vitro system, and they could then decide to not have to do an in vivo guideline study because it would most likely not give additional information. So I think that approach is how I see it would, and hopefully eventually we never have to go further than to the NAMs.
Any other comments on that? Let's move on.
Okay, I'll go with the next one. So, Alistair, I think this is in your direction, but there's probably a broader point here about chemical lists and how we benchmark NAM performance. So, did the high risk and low risk chemicals come from previously defined lists of chemicals? If not, and you developed these chemicals lists yourself, how were these chemicals vetted? In other words, was there a systematic approach that you used to develop these high and low risk chemical lists, what
Yeah.
specific variables did you use when determining which chemicals to include?
Yeah, so within that pilot study, I think what we did was we just, one question was around the reproducibility of the assays, particularly the cell stress panel. So we simply just took some chemicals from that initial cell stress study that we did, and then also selected some others that were used in consumer products. So there, it was just a selection, no particular process for selecting those chemicals. However, I do completely agree with the points that the author is making. And for the full evaluation, we've exactly tried to avoid the sort of bias in terms of how we select the chemicals. So what we've done is taken the full list from a number of these sort of chemical lists that you get from, say, Toxicological, and then selected them on the basis of, for example, is there actually an exposure scenario you can associate with those chemicals? Could you also associate a risk classification with them, so to do the benchmarking? But then from that, we also then took a randomization process in terms of how you select them. And that randomization process factored in both the use case scenarios, so getting a range of different things like cosmetics, agrochemicals, etc., drugs. So you got a full range of different use cases, chemistries, potencies, but again, doing it in a randomized way, so you're not really biasing then the full evaluation in one direction or another. Just to say that the reason for doing that is there's always a concern if you have people hand selecting chemicals, there'll be a tendency for people to choose their favorite ones, and they tend to be the most extreme examples, either the least potent and least ones of concern because they're the ones that they know, or the ones that are the most dangerous that just would kill all the cells, and actually from a sort of assessment point of view, they're the easy ones in a sense. So you wanted really a range. So that was, yeah, so we did put a lot of thought into that. It is definitely important.
Thank you, Alistair. Has anyone else got any thoughts on best practice when selecting these evaluation chemical sets?
Okay.
I think everyone feels the same way I do about that process. It's always painful. Back to you.
Okay, we have a follow-up on one of Anna's comments from earlier. Is it true that many global geographies are hesitant to accept non-animal alternatives? That puts the chemical companies, many of which are global, in a position of performing one assay which will be accepted globally versus two studies. With the growing costs of new chemical tox screening, the route of lowest cost would be in vivo route. So the question, what can regulatory agencies that accept NAMs do to educate or advocate for NAMs to regulatory agencies or countries that do not?
I was reading the question because somewhere I got lost in your description, so sorry for the delay. This is a really complex problem, and I don't know anyone who has really the answer. I can tell you the strategies that we've tried, one of the things that I always say is that, leaders have to lead, and that some organizations have to go first. So whether it's an individual company trying to do something first, or whether it's a company or an industry who's trying to advance things, someone has to go first. And some organizations are slower at adoption and some are faster at adoption. And sometimes it depends on the situation. Like, there's situations where EPA is really advancing some things, but we've been slow at other things, and vice versa, that some of our Australian and our Asian and our European colleagues have been a lot faster at adoption, and some, maybe North America is a little bit behind. And so some of it is just really just patience and tenacity and a commitment to change. And some of it is process that, that we really need to use the OECD. It really highlights the need for OECD to move faster and to really start to push the advancement of some of these frameworks that are adopting different approaches to guidelines that move away from the slow, expensive, not fit for purpose 3-ring trials. And that is my bias. I don't think that approach is really answering our regulatory questions. Some of it is working collaboratively, like one of the things that we've tried to do both at EPA, but at ICVAM itself, is to collaborate with our international partners, whether it's bilaterally, particularly, let's say, the US and Canada, or the US and EFSA or ECHA, or more broadly at NICEATM. So NICEATM is the International Corporation of Alternative Test Methods or something like that. I got the C wrong. To collaborate internationally to advance some of these topics. But there is no simple solution, because different regions and different countries have different regulations. Some are really anchored legally and statutorily in the OECD guidelines. Some are not. Some are just more open to bringing in new methods, and some are not. And it even varies within agency, like even within my own agency, different offices have different views on this and are moving at different rates. So there's no simple solution. It's a very complex problem, and one we just have to sort of working on and working, as a community on. It just takes time. Sorry.
Any final thoughts on that, Susanne, Amanda?
Yeah, I think the world is really changing in this respect as well. So I think during the last 15 years, there have been quite some achievements, and also the awareness, not only from regulators, but from the whole community. And I think it is also agreed upon that we need a change in the procedure to get more guidelines adopted quicker, but also change the validation process, because yeah, the world is changing. Toxicology is really also changing as well.
Thanks. Yeah, with that, I wanted to thank everyone for joining today, for our speakers, panelists, and our audience, of course. The recordings and slides from today's webinar will be posted shortly after we end today. You'll receive links to view the archived materials in your post-webinar email from Zoom. But with that, thanks everyone, and have a good day.
Thank you. Bye-bye.
Thank you.
Thank you.